Thank you, Liana, Chris, and Giovanni for the invitation to present our data virtually. It has been a challenge to keep us connected professionally, and you indeed deserve a big thank you for this stage you created in order to keep the Sphingolipid community vibe. I just close my... All right. So, the work that I'm going to present was performed in Tony Futterman's lab. But apart for Tony, the person who actually initiated the whole project was Dr. Rafi Schiffman, an active MD who meets Gaucher patients every day. Rafi thought that we are not still there in respect with the available Gaucher mouse model, especially the neuronopathic variants of the disease, and triggered the project actually. The funding was mainly by the Children Gaucher Research Foundation, and at some point we actually thought to name the mice after their foundation, because they really escort us along the whole project. Of course, there are many more people which I will mention through the talk, and in the end, acknowledge them. Most of the work that I will discuss today was recently published, but I will discuss also some extra things that were not published. So let me try to introduce the important terms for my talk through a quick historical overview on Gaucher disease. So Gaucher disease is the most frequent sphingolipid storage disorder identified by Philippe Gaucher in more than 150 years ago. And 20 years later, Gaucher disease was recognized as inherited disease. 30 years later, it was shown to include also neuropathic variants. Agion identified the GD hallmark, which is accumulation of glucosyl ceramide, and Brady in the 60s actually found out that the enzyme, that the defective enzyme is a glucosyl cerebrosidase, gluxurase, and also found out with colleagues that it's a lysosomal storage disorder. And the first mouse model came in 1975 as a chemical induced model using a CBE inhibitor of gluxurase. And when injected to young mice, it showed phenotype similar to neuropathic form of the disease. Many years later, when gene targeting technology was available, the GBA gene was knocked out, but these mice died shortly after birth. And around the same time, Genzyme developed the enzyme replacement therapy, first as isolate from placentas, later as a recombinant protein, and the substrate reduction therapy, which is basically inhibiting gluxur synthase to lower the burden of the lipid which is accumulated, came many years later. Interestingly, Alan Sidransky and Mistry found a link to Parkinson's disease. They found out that many Parkinson patients carry a mutation in the GBA gene, and many GBA heterozygous carrier develop Parkinson's disease. So it kind of opened the field to many more interesting directions. The IPS of all three types of Gaucher disease was established by a Feldman group about 10 years later, allowing some studies and therapeutic assessment in vitro. And during the last decade, there was a great improvement in the development of SRT using small molecules that can penetrate the blood-brain barrier. Until now, actually, there's no therapy that can penetrate the blood-brain barrier. It's all in clinical trials. And also gene therapy is now in trials. Today, I will present you our efforts to develop a new Gaucher disease model that we believe add an additional important tool to study this complex disease. This was not an easy task since Gaucher is a complex disease and phenotypically very, very diverse. Physicians have divided the disease roughly into 3 types. Type 1, which is a non-neuropathic form of the disease, although we know now the link to PD, and the neuronopathic form of the disease, type 2, which is a more severe one, where babies usually do not reach 3 years of their life, and type 3, which is if diagnosed and treated early enough, patient can reach to their 30s. There is about 300 known mutations in Gaucher patients, but correlation between genotype and phenotype is not high, which suggested the involvement of modifier genes. So this aspect was actually studied in our lab some years ago, where some putative modifier genes were suggested to influence the course of the disease. We have introduced Gaucher disease in many different wild-type strains by injecting the chemical inhibitor of the enzyme. And we realized that there are basically 2 types of phenotypes: mice that live short and mice that live long. This could be also analyzed by GWAS. We found a list of putative modifier genes, which fell basically or nicely between these 2 groups, and we are currently evaluating the role of these genes in Gaucher disease. Many efforts were aimed at the generation of the preclinical models for the 3 human Gaucher disease for type 1, 2, and 3. However, in many of these models, either the mice died very early after birth, or even when certain mutations that lead to rather milder forms of the disease, they cause death as well. Another problem which arose is mainly in type 3 models, where the accumulating lipids could not be detected, and only when additional gene in the pathway was knocked out, it was realized. So human and mice obviously differ, and the generation of accurate model remains a big challenge. Thus, we came up with the idea that although genetic background is crucial, however, if we use the same mouse, the same genetic background, where we can control the level of glucoceras and theoretically, we can basically generate all 3 types of Gaucher disease. If we shut down the activity completely, we might get a severe disease. And if we close the tap a little less, we might get type 3 or even type 1. That was, in a way, our naive assumption. And the system we used was as follows. We generated a transgenic mouse by inserting into the ROSA 26 locus a cassette that includes a trans activator under the intact promoter of the ROSA 26, which is not a very strong promoter, but it's continually expressed. This transcription factor can bind to a promoter upstream to a GBA gene, a cDNA, and the idea was to cross this mouse onto a complete knockout mouse via crossing it twice through a GBA heterozygous mice. And we managed to generate a mouse which is viable. The GBA knockout with a transgene expressing is a viable mouse as compared to the GBA knockout, the complete knockout. However, they were born a bit smaller, as you can see, compared to a GBA wild-type mice, for example. But this small size was true also for the other background, so also to the GBA wild-type and the GBA heterozygous. So mice that expressing the GBA transgene are born a bit smaller. We thought in the beginning that it's due to the fact that there is 10 times more expression of the enzyme, the transgene. However, when we did the lipidomics, we saw that Gaucher is not significantly reduced, which means that we still don't know why it is they have this smaller phenotype, this metabolic phenotype. So these mice at the age of about 1 to 1.5 months were given DOX in high concentration in their CHO diet. And what you can see that upon DOX upon DOX administration, the RNA of the transgene completely was reduced, both in brain and liver, as well as the protein. Here, for example, is the GBA wild-type mouse. This is with a transgene, and after DOX administration, it's going down. We did notice that the knockout background always gives a bit more protein and RNA. That was interesting and probably a compensation for the lack of endogenous GBA. However, despite the undetectable level of RNA and protein, we still saw glucocerebrosidase activity. This activity was about 30% of the wild type and about 50% of the heterozygous mouse. Assuming that the knockout, the GBA minus activity is 0, it was hard to go down completely compared to a wild type, which carries a transgene, which was almost, after DOX administration for many months, was almost like the GBA++, as well as a heterozygous. It was true for both brain and liver. And that led to accumulation of about a mild accumulation of Gaucher, about 4 times of the wild type, however, not in the liver. We also saw some glucose sphingosine accumulation, also very mild, as compared to the acute model where you see 100 times more glucosyl sphingosine. This is really mild. In acute model, there is about 10 times more. Here we see only 4 times or 5 times more. Okay, so as you can see in this survival curve, when dox is given at about 1.5 months, around 6 months, the mice start to decline compared to the legitimate control, which have the transgene or the other controls. When we look at the mice at around 4 months of age, before they have any overt symptoms, we do see in behavioral tests that they have some movement deficiency in the rotor rod, for example, or in a swim test. Here you can see, I will run it fast, a swim test. You put the mice in a warm box with water and you let them swim a bit. This is, for example, a mouse which has a transgene, no difference from the heterozygous mouse. They swim very happily. However, when you put the knockout mice, they tend to float more than swim. From time to time, they swim, wake up, and then float again. However, their circadian rhythm was not changed. So this means that they might have some neurological defects, and indeed, immunohistochemical analysis show that they have gliosis in the brain, in this case, microgliosis, especially in some region like the VPN, VPL, and the thalamus. This area here called the superior colliculus, which is important for eye movement. This is facial and oral cavities region. We see a lot of gliosis in the brainstem and in the cerebellum. When you compare it to an acute model, you see many more regions which were affected, and especially the layer 5 in the cortex, which you don't see in this chronic model. In addition, you see loss of neurons. This is a neo-N staining in the VPL region here, and you can easily see that there are much less neurons. Similarly, we see this is a staining of the cerebellum. You see in the control intact Purkinje neurons, while in the chronic model you see many lobes that lack Purkinje neurons, but not all of them, which was very interesting, only some of them. This is also seen in type 3 Gaucher patients. To better identify where exactly is the deficiency, we did a whole mount of the cerebellum staining using light sheet microscopy, and we could exactly define where are the deficiency in Purkinje, and we found out that it's mainly in the vermis region. However, there were few intact lobes, like lobe 2, 9, and 10, which is different from other lysosomal storage disorder, for example, Niemann-Pick model. So that is very interesting. We also performed RNA expression analysis on whole brain, and you can see clearly that the PCA show that they are apart from all the other controls. We got very few genes which were actually differentially expressed, which was very surprising. Only 59 genes that went up or down. You can see the control are very similar, no matter if they have the transgene or not. And this was very different from what we saw in acute model. There were thousands of genes change in the whole brain, while we had very few genes in the chronic model. And this might either be due to the fact that we masked the huge differences. And if you would take only specific region, we would see huge differences. But it can also be that that the mouse, which is chronic, is trying to get to homeostasis. And in the acute model, where mice die very early, they die because they have such a huge change. This is just using a published single-cell RNA-seq data to see if we can assign the changes of the gene expression to specific cell type. And here you can see that many of the genes that went down actually were in the cerebellum, in Purkinje neurons, and in other specific neurons. You can see these patterns. We don't know exactly what are the neurons, but we need actually to isolate specific regions, specific neurons to know more. Now we actually isolated one cell type, microglia, which is rather easy to isolate, and looked at this cell type to see if we see differences. Again, all the control fell in the same together, separated from the sick animals. You can see it also in the heat map. And again, the microglia of the acute showed a lot of differentially expressed genes, while the chronic model showed relatively low amount. We also saw that that the gene show certain enriched pathway in the acute model. It's mainly interferon genes, mitochondria genes, cell division were enriched, while in the chronic model, it was mainly nitric oxide signaling and cell cycle arrest. So again, we can claim here also that there is much more gliosis in the acute model, and we kind of mix the microglia with non-activated microglia, and that's why we got very few genes, but it needs further analysis. This is finally to show you that a part of the brain pathology, we also observe bone deformities in these mice. The most obvious phenotype is the gibbous, the kyphosis that you can see, which is starting pretty early. And this is also something one can find in type 3 Gaucher disease patients. We also saw reduction in their size, even when they start similar toward the end stage of the disease. And this is mainly because probably they stop eating at some point. This is just to show you how the mice look. You can see the kyphosis easily. They have some grooming, weird behavior. And they have a stiff tail. And they also have, let me go, clasping and basically showing some ataxia-like, of course, behavior. In addition, they also have skull deformities. There is like a tilting of their nasal bone, and in some cases, teeth are enlarged. This is again something that can be found in type 3 Gaucher patients, as well as thinning of their cortical and trabecular bone. Again, something that can be seen both in type 3 and 1 patients. So, to summarize and showing some open questions, the GBA knockout transgenic mice upon DOX administration exhibit chronic disease They live up to 10 months with relatively low increase of gluxer and glucosylsphingosine, leading to CNS and bone pathology, similar to type 3 Gaucher disease. In the CNS, the mice develop gliosis in the thalamus, midbrain, and cerebellum with neuronal deaths in specific regions. Mice develop kyphosis and facial deformation and exhibit thinning of cortical and trabecular bones. Gene expression show low number of changes as compared to an acute mouse model. The system, by the way, is reversible. Stopping DOX will allow increasing level of transcription again, and does evaluate the chance of curing existing pathologies. And the study of GBA expression level during embryogenesis, the transgene expression, by the way, is conditional. I didn't show you this, but you can go to the publication and see this. You can cross these mice with different Cre to generate this mutation in different cell type. We used a whole, a full deleter. And the selective vulnerability, why some Purkinje neurons are protected and other not, is basically our next task. So this is just to acknowledge everyone that contributed. Tamar, Shani, Ayelet, Natalia, and Sumin from Tony's lab, the veterinary resources, many of them, biological core facility, bioinformaticians, and of course, Al Merrill. Without him, we wouldn't do all this analysis. Samuel Kelly, who worked with him, Rafi Schiffman, and we got the vector, actually, the original ROSA 26 vector from Miyazaki in Japan. And thank you very much for your attention.